There are two kinds of students who study membranes. One memorizes the diagram and hopes the question is kind. The other actually understands what's happening, and those students can answer questions they've never seen before. Let's be the second kind. So I've taken one of my lesson slides here, and I've turned them into these interactive HTML files, which is just, like a nice scrollable file for you to be able to review in your own time. I'm going to talk through it, but it's linked in the description below, which will take you to my site, and then they're there for you to use. So when we think about cell membranes, now we need to think about a few particular key takeaways, some big ideas. So like most of the membrane is nonpolar, and that's regulating what goes in and out of a cell. It's super, super thin. It's just 10 nanometers thick. And a really nice visualization of this is when you look at red onion cells under a microscope, and you can see that you've got the outer cell wall that is rigid, it doesn't move. It really illustrates that point perfectly, that why do plant cells have a cell wall? Well, it's to make sure it adds that structural integrity. And then when a cell becomes plasmalized with the red onion, you can see it pulling itself away really nicely. And this is where we can draw back to our knowledge about osmosis. So this is obviously gone into a hypertonic solution. That means there's more salt molecules or more molecules in the solution outside than inside the cell. So water will leave, it will move down its concentration gradient, and we will end up what we call a plasmalized cell. Or if this was an animal cell, there wouldn't be any cell wall to hold that shape, so it'd end up doing what we call crenate. Now, the opposite of that would be hypotonic. That's where we end up with turgid cells. And that's where the cell membrane is then pushing against, and it's stopping it from bursting. If this was like a red blood cell and we put it in distilled water, you can imagine it would burst, and that would be what we would call lyse. So here is a really nice way to think about the fluidity of the membrane. It's able to expand and fill this void, and it's able to shrink back down again, and it doesn't break, mainly because the cell wall's there. So what would phospholipids do in water? This is a really nice way to think about why does a cell membrane form this structure that we know about. Now, if you drop 5 phospholipids onto a water surface, how do you think they would arrange themselves? So just take a few seconds to think about what would happen. You can pause the video if you want. I'm just going to click to see. Now you can see the heads have positioned themselves in the water, and that's because they're hydrophilic. And the tails are sticking out the top because they're hydrophobic. Now, let's think about if we added 20 that was submerged, what would happen then? Pause the video and have a thought. Now, what you can see here is and you can imagine this in here is now just like an empty space where it doesn't exist. It's going to form some kind of circle because ultimately the pressure is going to be equally distributed. But the idea is that all the hydrophobic tails are coming into the middle. You wouldn't have water inside it, but all of the hydrophobic tails are coming into the middle, the hydrophilic heads on the outside. Now, they're not attracted to each other, apart from the normal London dispersion forces, which is those weak attractions between any molecule, but they're arranging themselves like this because of this hydrophobic and hydrophilic property. Now, if we took 40 phospholipids and dropped them in, and water's on the inside as well as the outside now, you can see what we end up with is what we call a bilayer. Now, there'd be a bit more spacing here, but what it illustrates really nicely is why we get this cell membrane. There wouldn't be the overlapping of the tails, but what this little animation does really get across really well is that we get a bilayer. And we're getting a bilayer because the cytoplasm is predominantly water. So in our cells, they're going to naturally form a bilayer as well. Now this looks like a little vesicle, which is what we've encountered when we talked about cell biology before, when we named all the different parts inside the cell, and this is going to help with compartmentalization. Now, let's keep going. So the bilayer is just a scaffold. And what do I mean by that? This is what we call the fluid mosaic model. There's some cool experiments to show just why we call it that, like namely where you can tag proteins. And what you see if you tag proteins with phosphorescent dye is that they will move. And they will move randomly over time. So that's where it gets this idea of it being fluid and a mosaic, like a picture mosaic. We've got proteins that move around each other. They could be, a channel protein for facilitated diffusion. They could be, you could have a protein pump. All of the ones that span the membrane, we call them integral. Ones that hang out on the outside are peripheral. And what they've got in common is the bit that's embedded is also non-polar. Now, you probably spotted a mistake here with this diagram in that fact that it's only got one tail here. So we've got the hydrophilic head, and there's only one hydrophobic tail. So, I've made this interactive HTML with a nice little skill that I've been developing with Claude. So there's a little bit of feedback for him there. Not quite perfect, but fortunately you've got me talking you through them. So one thing you'll notice is most of the membrane is non-polar. And what that means, if we keep scrolling down here, we've got the roles of the proteins. That's something you can go and reference. I'm not going to talk through that right now, but because one of the most important things that I'd like to just skip down to is about what can move through the membrane. Now, most of the membrane is non-polar. Once again, messed up the tails. Not a big deal, but I'll be telling them off later. But most of the membrane is non-polar, so small non-polar molecules can diffuse freely through it. Things like oxygen and CO2. Now, this is polar. It's also small. Water is a small molecule. But the difference between these guys is that this is polar. It's got a positive and two negative, like tails with the hydrogen. It's got the negative charge of the oxygen because the electrons are more attracted to it. And then the hydrogen has got this dipole where they are positively charged because their electrons are more attracted to oxygen due to its stronger electronegativity. Now, they will only move through the membrane slowly because most of this is non-polar. Now, if you're large and you're charged, you don't stand a chance. You need something to help you move through. So you need a protein carrier. Now, we'll skip back up and we'll just think about cholesterol. So one of the key takeaways here is that cholesterol will modulate the fluidity, or we could say it buffers the fluidity. But what that means is that it will increase fluidity or decrease fluidity, but dependent upon the temperature. And this little table summarizes it really nicely. You can see here, so cholesterol is what we call an amphipathic molecule, just like water. Just like the phospholipid. Now, a phospholipid here, once again, should have two tails. I'll update the one that's on my site to make sure that you've got a really nice reference point. But luckily, you've got me guiding you through this. And the idea is that this is also an amphipathic molecule. They're both amphipathic. That means they have a hydrophilic area and a hydrophobic area. And it's going to embed itself in between the phospholipids. Now, what that means is that when it's cold, when it embeds itself, it disrupts the tight packing. It disrupts the London dispersion forces between the adjacent phospholipids, and it's an increased fluidity. But if it's hot, then it actually pulls these phospholipids that are trying to move away from each other because their kinetic energy is increased, and it pulls them closer together, and it decreases fluidity. So it's what we call a modulator or buffer for fluidity. Now, if we put that into some context and we think about a crocodile icefish, now, one other thing that we want to know about before we fully think about it is that what else could affect fluidity? Now, if you've got a crocodile icefish and we just think that it's living at -2 degrees, well, first of all, we know that if it wants to increase the fluidity, it's going to have to add cholesterol to its membrane. Now, what else could influence this? So saturated or unsaturated, that matters. So what I mean by saturated is that there are no double bonds in the fatty acid tails. If they are unsaturated, they become kinked. And you can see here that kinking means that there's an increase in space between the phospholipids. And a kinking is caused by the unsaturated fatty acid. Now, it's only in cis unsaturated fatty acids. That means that the double bond, the carbons either side of that, are on the same side. You have the other type that we've spoken about before when we looked at biological molecules, called trans unsaturated fatty acids, and that's where the hydrogens on the carbons are opposite sides. The way I remember it is that, you know, if I make that double bond with my thumbs and I turn my fingers to one side, then it's going to show me the letter C. And if it's cold and fluid, it means that that kinking is going to increase the fluidity when it's cold. Now, this crocodile icefish lives at -2 degrees centigrade. Now, what would you most likely find in a crocodile icefish membrane compared to a warm water fish? We have a couple of seconds to think. And what's lovely about these interactive HTMLs that you'll find on my site is that if you click one, it's going to give you some feedback. And if we clicked the wrong one, so more unsaturated kinked fatty acids, you might also find some cholesterol. But if I'd clicked the wrong one, then I would have got this feedback too, and gives me a few tips in there too. Now, we already spoke about what moves through the membrane. It's really important to remember that small non-polar molecules like oxygen and carbon dioxide, nitrogen gas, they can move straight through the membrane. There was a question this year in the IB paper looking at a steroid. So steroid hormones, they have those 4 distinct rings, and they are able to also pass freely through the membrane, and that's because they're non-polar, and most of the membrane is non-polar. So this is one of the big ideas. So most of the membrane is non-polar, small ideally, but also larger ones like steroids, they can pass straight through the membrane because of, in this case here, they're not going to repel each other. Whereas something that is charged, like water, it can go through slowly. That's why you have to leave your osmosis experiments for 20 minutes to an hour or something, but it takes a lot longer because it has that polar charge. And if you are large or an ion, you don't stand a chance. Maybe you squeeze through every now and again, but ultimately you need a protein carrier. And those protein carriers, you know, they're the ones that have to go across the whole membrane, so we call those integral or transmembrane proteins. Now, when you go and click on the file later and have a little look at the HTML, you can see there's also some questions. I recommend you practice first, and then you can click for the mark scheme. Right, I hope you've enjoyed today. There's also a little, I've stuck in a close at the end, just to summarize. So I hope you've enjoyed. This is the first video that I've recorded in these series of Biology by Bradford. But there's going to be lots more dropping. I'm going to try and do one a week, just picking a few select topics at first that I know students struggle with, that addresses the key concepts. But this video, although made for, you know, IB biology at the forefront, I'm also going to be making ones for AP, and where I can, I will make sure that they address both of the syllabuses. Okay, thanks for listening today, and hit like and subscribe. And in the comments, feel free to drop any ideas of things that you would like me to explain in the future, and I can focus on those topics.